Welcome with this inaugural lecture from Professor Heike Grabsch. For people who are not that often in the ceremonies, you see different clothes that we have. That means that people come from different places, and in this place even international different universities, so that's a first honor for Heike, that these people all took the effort and came to join the ceremony. I think we know already what we have to do. We should keep calm and listen to a pathologist. Professor Grabsch.
Yeah, dear valued members of the board, colleagues, friends and family, I'm very delighted that you all have come. And after this wonderful morning, you now have to listen to me for 45 minutes. This is why I've put in there, keep calm and listen to a pathologist. And if people asking me, what are you actually doing nowadays? And then I say, well, with pride, I say I'm a pathologist. And actually, I'm a histopathologist. So people who now think actually of these type of TV series where they're people dealing with dead bodies only and looking for crime scenes, this is not what I'm doing. I'm a histopathologist and don't deal with crime. So I look at cells and tissues from living patients to make a diagnosis, to guide patient management, and for research purposes. And you can see here below a typical picture of a histological slide of a gastric cancer. The normal mucosa you can see on the left, and then you can also, with no special knowledge, you can see that on the right there's something wrong. And this is what I'm looking at every day. The other, I think, very important task we have as a histopathologist is that we are a core, or not to say a key member of the multidisciplinary team in the hospital. And you can see here on the big screens, there's a pathology slide next to radiology, and we are actively involved in guiding the treatment of the patients. And of course, we have surgeons and oncologists and all the other members of the team here. But I haven't been always a histopathologist, and many of you may not know. So in the 1980s, I actually started my professional career working in the oil refinery in Hamburg. And you can see me here on the top of the platform, fully geared up and with the helmet. And you wonder then, okay, how do you get from there to become a pathologist? And is it actually so different what I'm looking at now? So in an oil refinery, you can see here on the left-hand side a simplified flow diagram of an oil refinery, and there you have the crude oil coming in. There's the crude oil coming in here at this part of the refinery. There are all sorts of processes going on which are highly regulated. And then at the other end, you have diesel coming out and you have gasoline coming out. And everything is highly, tightly regulated and needs to be controlled. And this is what you can see here in the operator room. And if it's not controlled, can be such big disasters like here in 2005 in the BP Texas City refinery. And look at this now. Now, in 2016, I'm again looking at pathways. And I'm again trying to figure out what's going on. How is it regulated? And this is here, in this case, the EGFR pathway. And what we can see here, the EGF is coming in here. But what is the big difference now that we're looking at cancer or how things are regulated is that we have much less control over the process. In the refinery, there's a computer, there's a program which actually drives the refinery. In the pathways in the human being, it's much less known, and this is why we struggle. So now we're coming to some audience participation time, and also coming back to the crime scene investigators. And my first question for you is now, what do you think, what is the most common cause of death in a country like the Netherlands? Is it really like these crime scene investigating films want to let us believe murder, accidents, unnatural disease? Is it cancer or cardiovascular disease? So I'm now asking you to show up your hand. Who thinks the most common cause of death is murder, accidents, and unnatural? So nobody thinks that, okay. So who thinks it's cancer, the most common cause of death? Yeah, we have some show of hands. And who thinks it's cardiovascular disease? We have more for this. And actually, you're right. So the last people were right. The top 10 causes of death in 2012 in high-income countries like in ours is cardiovascular disease. It's not cancer. Cancer is quite common, and it's obviously quite well in the public domain, but it's cardiovascular disease. If you're looking at low-income countries for comparison, it's infectious diseases. So those people are really, cardiovascular disease does not play a role. But I actually don't want to talk about cardiovascular disease, although my first thesis was about cardiovascular disease, which I did in the anatomy department at the University of Cologne. Now I want to talk about cancer today. And first of all, how does cancer actually start? And so if you're first looking here at one of the factors, which is cell division. So in the top part of the slide, you can see the normal cell division. And if there's normal cell division, the cell has to duplicate its DNA content and separate it really accurately to the next two daughter cells. And if there is then some genetic damage happening, like here with this yellow arrow happening, and then the DNA gets damaged, then actually in all normal cells we have policemen around. And here this is policeman P53. And this policeman actually makes sure that whenever there is damaged DNA that cannot be repaired, these cells are dying. So they are sorted out and they can't proliferate. But if that policeman isn't working, is sleeping or doing something else, then actually these cells can proliferate. And you can see this here. And they can proliferate completely uncontrolled until a big tumor arises. And there can be more damage on the way, and this is why they have different colors. So you can instantly, I think, envisage that the more cell divisions you have, the more likely it is there is some damage, and the more likely it is that cancer will develop. And that actually explains why the cancer incidence increases with age. And you can see this here, so the age is at the axis, and here you can see the rate per 100,000. And it's actually true for male and female. So keep this in mind, the cancer incidence increases with age. Then the other evidence is that cancer incidence also increases with height. If you are very tall, you need to have had more cell divisions. So you have had a higher chance of something going wrong. And this is here shown in a very big study, prospective cohort study of more than 1 million, in this case, women in the UK. And you can see that there is almost a linear relationship here in this graph between the height and the relative risk to develop cancer. And it starts about sort of if you are taller than 1.55 meters, then you're running an increased risk. So now the next question, considering that elephants are so much bigger than we are, so they measure 3 to 4 meters high, and they have much higher weight, and they have lots of more cells, but they have the same age than we have. And considering what I just said before, do you think that elephants die more frequently from cancer than humans? So if you think that this is true, can you please raise your hand now? Do elephants have more frequently cancer than humans? There's one hand, okay? Nobody else thinks that, okay? I found it quite astonishing. But yes, indeed, you were right. So although elephants are quite big, they're quite tall, they have a much lower frequency of cancer. So obviously the idea that the bigger you are or the heavier you are, you have a higher risk, that does only work within a species. So if you compare different humans, it works, or if you compare different elephants, but it doesn't work out between elephants and humans. And then just to finish that off, you may wonder, what about the mice? And actually here on this graph, you can see that the mice, which is obviously a very tiny animal, has the highest chance of developing cancer per cell they have in the body. And the whale, which is quite old, the whale can be up to 200 years old, very heavy, so it really has two risk factors we have in humans. The cancer incidence is extremely low. So the question then is, can we learn something from this? Can this paradox help us to learn for human beings? Well, there have been now really recently in the last year data coming out about elephants and elephants' DNA being sequenced and copy numbers being investigated, which I think is quite fascinating. And so experiments have been made where you take blood from an elephant and you radiate the blood from the elephant and you radiate the blood from the human being. And what is happening when you radiate blood is that the cells get damaged. And then the damaged cells should be getting apoptotic, so they should die when you damage them. And what you can see here is, in the blue bars, so in the darker blue bars, is that the lymphocytes from the elephants, they are dying much quicker. So they are much more able to react to the damage, and they will be much better eliminated than from the humans. It's almost double. So if you're comparing the dark bar with the lighter blue bar. And then even more so, I think what I found fascinating is elephants actually have more copies of P53. You remember the policeman who is policing in our cells, so they're not only having 2 like we as normal human beings, they actually have 40 of them. And that is considered to be one potential cause why elephants have this better damage control. And you can see here in this graph, it's almost a linear relationship. So the more P53 policemen you have in your cell, the more lymphocytes are actually dying when they're damaged. And then there are people who have a mutated P53, only 1 copy, and they do really badly. So those lymphocytes are actually not reacting to the damage. But let's go back to humans. We come back to the elephant a little bit later. So now comes my next question to you. We're looking here now at Napoleon Bonaparte, and we're looking at John Wayne, both here on these pictures. What do these 2 men have in common? Has anyone any suggestions? Yes, there's somebody obviously guessed that because I'm talking about gastric cancer, this is what they have in common. And yes, indeed, apart from both loving horses, they both had gastric cancers. But why am I picking those 2 out of the 700,000 people who die from gastric cancer every year? I'm picking them because they are very good examples of 2 different causes of gastric cancer. So if you're looking first at Napoleon Bonaparte, he had actually hereditary, so inherited, diffuse type gastric cancer. This is something you develop because your father or your mother had already the disease. And as you can see here, this is a pedigree. So this is the father, this is the mother, and if it's red, then that person died from the disease. And this here is Napoleon, and you can see that also sisters and brothers also had gastric cancer. Overall, it's very rare, so less than 3% of all gastric or stomach cancer are actually falling into this category. Usually people are very young, 40 years old when diagnosed, or even younger, so they can be teenagers, and they usually have an Ekaterin germline mutation, although I could not find whether actually this has been sequenced for Napoleon and his family. And what is important clinically here is that if you're having such a germline mutation, you can also develop other cancers. So those people require genetic counseling, and they require then potentially prophylactic surgery. So what about diffuse type gastric cancer? So why do we call this diffuse type gastric cancer? And I think this picture just illustrates how a cancer grows and why we call it diffuse type. So just to explain this, I've used a special stain to stain all the brown epithelial cells. So it does not discriminate between normal and cancer, but I think you can see here, so this is a normal, still a normal mucosa, normal architecture. All these areas here, they are normal. And then you can see here something, lots of brown dots, sweeping through the mucosa and pushing themselves into the wall of the stomach. And all these small brown or black dots, they are cancer cells. And because of this growth pattern, this is why we call it diffuse type gastric cancer. So let's look at John Wayne. So John Wayne probably had an intestinal type gastric cancer. And because it's not inherited, this is why we call it sporadic. So if you're developing the cancer and nobody in your family has a mutation, that's what we call it. And I'm using that example, first of all, to explain how gastric cancer can also develop. So we just heard about the mutation, now about a different pathway. And this pathway has been named the so-called Korea sequence. So if you have an infection with the bacterium, which you can see here, this curly green thing, I'm really trying to find the mouse here. So it's the green organism there on the middle of the slide. So if you have an infection in your stomach, you develop a gastritis. This is what we call an inflamed stomach. And if it's going on for some time, the actual mucosa gets very flat and changes. And this is what we call atrophy. And then in the next step, it will be replaced by a different type of mucosa. And that's what we then call intestinal metaplasia. Further on, you get then dysplasia and adenocarcinoma. Also note that salt, excessive salt, is also a risk factor to develop gastric cancer. And on the left-hand side, you can then see on this pink bluish picture on the bottom that this type of cancer looks very different. And this is why we call it intestinal type and not diffuse type. And the top picture shows you this brownish material, and that's actually the organism, the Helicobacter pylori, as we can see it on the slide. So I don't know what type of treatment Napoleon received. Probably none, I would guess, because the first gastrectomy wasn't invented at that time, and there was no chemotherapy. And what we know is that John Wayne only survived 3 years. And probably because he was in the US, he would have had surgery, and most likely after surgery, some chemotherapy and some radiation. In Europe, we do it a little bit differently. So in Europe, we give chemotherapy first, then you have surgery, and then you have another round of chemotherapy. And I've put this red box around surgery because I know that we have some good upper GI surgeons here in the audience. And I wanted to look at who actually did the first gastrectomy. But not only did Bill wrote the first gastrectomy 135 years ago, he was an extraordinary man. And because he actually had 3 professions, so he was really crossing the borders. So he was a musician, and he played piano and violin. He composed some music which you can actually listen to over the internet. And he was also a conductor of a big orchestra. And then what obviously pleases me, he was a histopathologist. And he wrote several books and monographs about histopathology and competed with Virchow for a chair in pathology. It's not entirely clear whether he lost against him or whether he didn't take it up. If you're looking at different literature, you find different information. But in the end, Virchow took the chair, and Billroth decided to become a surgeon and take on a chair in surgery at a very early age, when he was 31. And if you're then looking for literature, he actually worked scientifically and very active in all the above areas. So he has written about music, about the theory of music, he has written about histopathology, and he has written a lot of surgical books. And he then obviously is famous for the Billroth 1 and Billroth 2 gastric cancer surgeon. So let's cross some more borders. So we've seen for Billroth it was really beneficial, and he actually said art and science should not be separated, and that all scientists need to have the same imagination as artists. But let's be some more practical and look at the comparison between gastric cancer, small bowel cancer, and large bowel cancer. So these are three different parts in the gut we have, and so first comes the stomach, then the small bowel, then the large bowel. And the yellow part is at the top is the relative length of the small bowel. So it's about 75% of our gut length is made from the small bowel. But then, we only have 2% or less small bowel cancer. So how can this be that in this huge surface we have there, you have such a low incidence? Whereas in the large bowel, which is only 1.50 m maybe, we have more than 60% of all of the cancers. And so we were intrigued and thought, let's have a look whether there is some biology here. Do they actually have the same molecular phenotype? Do they behave differently? Most importantly, also to provide some guidance for the clinic. How should you treat them? Should you treat small bowel cancer like gastric cancer? Or should you treat it like large bowel cancer? So we did some work on this. And that was actually done with Gerrit Meyer and Bauke Oelstra, who are both in the audience here, and was one of our very early work where we started to compare the copy number profile and found that actually the small bowel is much more like or closely related to the lower GI tract than to the upper GI tract. And that obviously may inform clinical treatment. Then the next two studies I wanted to briefly introduce to you, which are also crossing borders, they were driven by the fact that the gastric cancer incidence is extremely variable throughout the world, as you can see here. 75% of these cancers occur in Asia, 13% in Europe, and about 2% in Africa, and then obviously in other countries. So we wanted to find out if we compare those two populations, the molecular phenotype of the gastric cancer, is it the same or is it different? And the first study we did, again, together with the Amsterdam group, is we looked at African gastric cancer and compared them to UK gastric cancer. And this thing, what you call here, what you see here, this is a so-called heat map with lots of green and red dots. And that means if there's a red or green dot, something is not normal. And if it's black, then it's normal, there is no change. And then there is a computer program that then organizes the cases in a way that cases that are similar are grouped together. So the patients are coming into columns and the chromosomes are into rows. And what you can see here, what I want to draw your attention to is between the two big blue bars, you have much fewer red and green dots as if you compare it to right or left. And interestingly, these are the gastric cancer of the Black African population. And so that study told us, they are not the same. It's not just the incidence, there may be some biology in here. So we also thought, well, let's then also compare to Asia. And my collaborators are sitting in Japan and Singapore, and this is comparing the data from Yokohama and the UK. And you can see here, and this is, I have to explain, this is a so-called survival plot. And that means if you are on the top bar, where now you have the Japanese flag, you are surviving longer. And if you are lower the curve is, the poorer the survival. And what you can see here is that there is a significant difference between the Yokohama patients and the Leeds patients. And this is after already considering all the different height, different weights, the different types of surgery. All these factors were considered, and even then you still have a difference. And so we were really thinking, well, then there must be some biology in there. In the past, in the last decades, people always says, oh, it's the surgeon. The surgeons are just better in Japan, and this is why we in Europe having such poor survival. But actually, we found something else. So in a very nice multi-center study together with the Patrick Tan in Singapore and several other collaborators, we found that actually there is a gene expression of the T cells, and the T cells are lymphocytes in your tissue. So people who are trying to defend you against cancer. There are differences between Japan and Leeds, or between Asian and non-Asians. And you can see this here on the top on these bar charts. And you can see that where I've put the dashed box around it, that certain genes are just more prevalent in the non-Asian population than in the Asian population. And also, if you look at this here on the bottom, you can see that where I've used a staining for the same cell types, that there are more brown dots on the left side than on the right side. And that means we have more of the cell type in the Caucasian. So this was quite a landmark study, and I think it's really the first study that actually showed that there is a biological difference. It's not just the surgeon. It's not that the people are slimmer in Japan. It's really a biology phenomenon. And then we heard this already this morning, and I'm only showing you this very briefly. There are many, many molecular studies trying to do new classifications now. So we have made an enormous progress in gastric cancer over the last decades. And I don't want to go into detail at this time. But what I wanted to show you here is that actually the prognosis, the survival or outcome of the patient hasn't changed in the last decade. This is from a very recent trial we have performed in the UK. The outcome in 2016 is from the STO3 trial in gastric cancer. And then the green dashed line, which you can see also there, is from another trial that was reported 10 years ago, so-called MAGIC trial. And you can see that there is no difference. So actually, yes, we have made a lot of difference in the understanding of the cancer, in the biology, but for the actual patient, there hasn't been much progress made here. And so the question then is, what do we do then? We know we have now all this biological knowledge, but what is our next step? And I think it is really important that we see the tumor in a more holistic way. With all the studies in the past, they have looked at the tumor cells, and the tumor cells are here, these the blue ones. Here they are the tumor cells, and they are growing here into the tissue. But if you're looking there, the tumor cells are not alone. You have tumor cells, but you have all these other things here. So you'll have extracellular matrix, we call it, so some fibers, you could say. This is this material here. Then you'll have other cells with these elongated pinky or orangey ones, which are fibroblasts probably, or myofibroblasts. You have vessels, which are this mouse is really not working nicely. So the round structure in the middle with the hole, that's the vessel. And that vessel is supplying, obviously, the tumor. And then you have infiltrating immune cells, which are this green cell at the bottom, and also this more pinkish or lila cell at the bottom. So that the tumor is basically surrounded by all sorts of things. And at the moment, we're really not looking at that. We are just, we're characterizing the tumor cells. We know the tumor cell is proliferating. We know what the profile is. But hang on, what about the other cells? I think they must have a role here in this context. And I think we really need to set out for a neighborhood watch. So looking not just at the cancer cell, but really looking beyond that. What is there? Are there cells? Are there vessels? What are they doing there? Are they proliferating? Are they secreting something? Are they talking to the tumor cells? Or are they actually completely not doing anything? Because I think, like in real life, the tumor cells will have good neighbors and bad neighbors. And these neighbors will influence what the tumor cells are going to do. And I'm just showing you here on the top part, this is, you should see some red color and some blue color. And the blue color is, in this case, a staining for collagen. And then on the left top-hand side, you can see there's very little blue. And so there are a lot of tumor cells, but very little collagen. And then on the right top side, you can see that there is a lot of blue and relatively little of the red colors, which are the tumor cells. And I'm convinced there must be a difference here. I can't believe that this is the same thing and that these tumors are different, are behaving in the same way. And we have shown some initial work on this, that the amount of stroma is related at least to your prognosis. But that was just a quantification and observation. We haven't done any work onto understanding what is going on, why this tumor has so much stroma. But you can imagine that if you have a lot of stroma, you actually have really problems to get any medication to the tumor cells because there is no vessel to transport it. So we have already now started to look at some vessels. If you look at the bottom panel on the left-hand side, there you can see the tumor cells in blue, and then hopefully you can see that there are brown things going around the tumor. And these are the vessels here stained with an antibody called CD31. And what I found fascinating is that actually the vessels not crossing. They are just on the outside of the tumor. There's no vessel in the middle of it, and there's no vessel going through there. And so there must be some interaction between the two cell types. The tumor must in some way communicate, well, I have enough food, I don't need any more oxygen, the vessels don't need to come inside. And we need to look at that, and in more cases. And on the on the right-hand bottom, we have talked about this morning already, there's a stain for immune cells, and clearly these brown cells, they are sitting on the tumor cells, and they need to, I think, interact with the tumor cells. But we need to find out how and why and what are they doing there. So I want to make 3 statements now, where I think what is important to make progress for gastric cancer patients. I think most importantly, the treatment should be tailored to the characteristics of the actual cancer patient. And I want to emphasize that I think we need to look at the patient, not just at the molecular profile. Because if you're having a very frail patient, yes, okay, that patient may be HER2 positive, but that patient may just not be fit enough to get the treatment based on the molecular profile of the tumor. So we need to look at the whole patient in order to make progress. I personally am very much convinced that this is a multidisciplinary approach, and I believe, nevertheless, that we need to obviously continue looking at tissue and doing research and translate it into clinical routine. But what is very important is that we have well-defined, well-annotated cohorts of patients, ideally from clinical trials, or ideally or including epidemiological data. And I think here we may be at a place in Maastricht where that could actually turn out to be a reality. So when I was still in Leeds, which was 2 years ago, I've already started to realize, well, I need to find not just friends where I can travel to, but also some collaborators, people who have the same ideas, who have the same interest, and who are passionate about finding out what's going on and let's improve the survival of the people. And you can see them here, and I think most of them will be in the audience, which I'm really very grateful for. What we also established in Leeds, and you can see this here in the bottom left corner, is that we have collected all the major clinical trials in upper GI cancer, so in gastric and esophageal cancers, that were ever conducted. So we have built up the resources. So yes, I think we are very well equipped. But now that I'm here, I think I have really been, one can say, heartened or energized by the opportunities that now all of a sudden come to me to expand that network. So being here in Maastricht means, okay, there is no gastric cancer surgery here, but hey, Zuiderland, Sittard and Herlen are very close. And I found there are some very enthusiastic surgeons and pathologists who were started to work together. And it is the largest center in the Netherlands, so what else do you want? So we really are at a place where we can expand what we are doing. We have started now also to look towards Italy and with Franco Roviello in Siena and have done the first stainings and result. And I've been in Doha, and there we will be seeing, I think, some progress. And of course, Aachen is very close. And there we have already, in the 2 years, we have already published our first study together. So there's really action is going on here. And also being in Maastricht means I'm very close to Brussels, and because I'm very much involved in the ERTC, and that will make life much easier. And through the ERTC, we have now also very stronger links to Japan. And last but not least, I also have now established my first sort of industry connection with Definiens in Munich. So I really think we are at a good place here, and we can do it, and I'm convinced that we're gonna deliver on this. And what I'm also really like is that the team UGI has now 2 fractions. So we have a branch in Leeds, and we have a monthly conference on what's going on, what are the investigations, and we have a Maastricht branch. But we are part of the same team, and I'm extremely proud to be part and leading this team of very enthusiastic, motivated, and committed people. So we will make progress here, and they work also a lot for this event behind the front. So I need to thank you also for this. And I need to also thank the funding bodies which have funded me so far, and I'm still benefiting from that. And at this moment, I also want to thank the patients. Without the patients, and without the patients allowing us to collect their material, to collect their data, to analyze this, I would not be here. We would not be able to do any research whatsoever. So I think we should not forget the patients. And I would like to really thank the patients as well. And I need to obviously thank my husband, and I'm very grateful for supporting and joining me in this incredible journey and sharing my commitment 24/7, so every day, 24 hours, 7 hours a day. And Johannes, you may not realize this, but I've drawn enormous strength from you trying to build steam locomotives and from scratch, so that has been really a pleasure. And my father and other members of the family are here with us today, and I would like them to be recognized for their contribution to my career. I pledge that I will do all that I can to redeem the face of the College van Bestuur and the Raad van Bestuur from the university has placed in me, and thus with humility and really a burning passion, I accept this chair. Last but not least, I want to thank Phil Quirk. Phil, it is a true honor to stand here in front of you and having the same gown and taking up this chair of gastrointestinal cancer pathology at the University of Maastricht. You have been and will remain an outstanding role model for me, a mentor and good friend over the last decades, and I commend your excellent leadership. I wouldn't be standing here without your support, your guidance and encouragement, and so it has been really great. We have been here together in 2003 on a seaplane, and that was actually Phil who wanted to go onto this plane. I wanted to go on a helicopter, and he said at that time, well, the next time we're going to go to San Francisco together, we're going to go onto the helicopter. But we have both been so busy over these last decades that we actually never made the helicopter flight. So I think this is still something on our list to do. And lastly, this is now my last slide. I firmly believe, I'm very passionate that we need to give something back. I have been very privileged, I think, throughout my life so far, and I think we need to give to people who need something back. So the last year I went with my team on the 545 walk. We walked the 12 kilometers up and down the hills here in the region for the Cancer Onderzoekfonds. This year I'm going to do step up a bit, and I'm going to walk in the sand for a whole week. And all the money or what I can raise will go to actually improve pathology and laboratory medicine in an under-resourced country. So I've pledged that I will give everything what you might want to give me to, on the occasion I will give this to this initiative. And I'm extremely pleased that I have looked just up this morning that I've reached already 80% of the £2,500. So people have already been donating for me, and I think that's brilliant. There will be a donation box also during the party and in the reception, or you can donate online. And if you're saying, well, I don't have money with me, that doesn't matter. You can donate until end of November. So no excuse. And by that, I've spoken.
Professor Grabsch, thank you for the very interesting lecture. It was very interesting how you build it up, and that you also give some room for us to answer difficult questions. And very good that we were almost everybody wrong at one of the questions. So I learned a lot. Now we're gonna close this ceremony, but first we're gonna make a picture with all the professors on the stair, and after that we walk to the reception area. So you can follow us, but first a picture and then we go to the reception area. Thank you very much.